Hi everyone, welcome to this video presentations. In this part of presentation, we'll be discussing about microbial taxonomy. Taxonomy in general is the classification of organisms and for their easy understanding and easy identification. So for microorganisms also, we have to follow a certain system to classify these organisms. So here we will briefly describe what are the different strategies used for classification of microorganisms and how do we actually classify microorganisms. Usually the prokaryotic systematics or the prokaryotic taxonomy is a little complicated when compared to the higher level organisms because of their microscopic nature and difficulty for observation of different characters, and hence usually the taxonomists usually follow a polyphasic approach for the classification of microorganisms. And the polyphasic approach is nothing but different approaches are being incorporated, like phenotypic characters and genotypic characters together will be used for characterization. Also there will be molecular and ecological characters. So different forms of characters will be analyzed and the data is collected, and accordingly the classification will be performed. So before moving into the classification and their content discussion, we will have to see some historical timelines. See, the classification of organism has begun even at the time of Aristotle, and he classified organisms into plants and animals into two groups, right? And in the years thereafter, different scientists have introduced different concepts to classify organisms. And now we know that we usually follow the five kingdom of classification for living organisms, right? In 1730s, Carl Linnaeus introduced uh, a system for the naming. And in the 1730s, Carl Linnaeus introduced a formal system of classification in which usually the organisms are classified into plants and animals. That means the two kingdom concept that was introduced in 1730s. And thereafter in 1860s, Ernst Haeckel proposed the three kingdom concept, in which the kingdom Protista were added. And the kingdom Protista included bacteria, protozoa, algae, and fungi, the smaller group of organisms. Right. So they were taken out of the plant kingdom and included in another kingdom called the kingdom Protista. That was done by Ernst Haeckel in 1860s. And this concept is called the three kingdom concept. Then in 1960s, the new kingdom or a new group was identified as prokaryote, right? It was much well defined during that period. And in 1960s itself, Robert Whittaker introduced the five kingdom concept, in which the prokaryotes were placed in a specific kingdom called the kingdom Prokaryote or kingdom Monerae, right? And the eukaryotic organisms were comprised in the other four kingdoms. All right. And even after that, there is another type of classification called the six kingdom concept. So Robert H. Whittaker's is the five kingdom concept. That is what we usually follow. And even after that, there is a six kingdom classification system introduced by Carl Woese, in which the prokaryote has been split into two as eubacteria and archaebacteria. So these are the different timelines for taxonomy as a whole. So these are the major systems like the three kingdom by Haeckel, in which plant, animalia, and protista, and the five kingdom were Monera, protista, fungi, plantae, and animalia. Then the sixth kingdom, in which there is a eubacteria and archaebacteria in addition to the other kingdom. Then Carl Woos actually also introduced the domain concept, in which all living organisms have been included in three domains of life. One is eukarya, archaea, and bacteria. And these two terms we usually get confused. What is kingdom Monera and kingdom Protista? Usually, kingdom Monera consists of unicellular organism with a prokaryotic type of cell organization. That means these kingdom comprises prokaryotic organisms. So all bacteria belongs to kingdom Monera, right? In the classification of when it was in three kingdom concept, or in the three kingdom system, these prokaryotic organisms were included in the kingdom Protist, right? There was no kingdom Monera or other kingdom to cover these organisms, so they were included in kingdom Protista. And so what is kingdom Protista in five kingdom classification? When it comes to five kingdom classification, the protists actually cover eukaryotic organism, but these eukaryotic organisms are having a unicellular organization, right? Usually most of them are solitary cells, or cells lacking true tissues and their properties, right? They will have like properties like ingestive, absorptive, or phototrophic nutrition. So this is usually not a feature of other eukaryotic multicellular organisms, and hence they are included in another kingdom called kingdom Protista. And we have examples like certain algae, protozoa, and certain simple forms of fungi. Other fungi are included in the kingdom fungi, right? So certain simple forms of fungi and protozoa and algae are included in kingdom Protista. All right, so this is the difference between the Monera and Protista. And this is the Calvus concept of life for in terms of domains. This is There is a universal ancestor. From the universal ancestor, the bacteria and the domain eukarya was developed, and from eukarya or from archaea, the eukarya developed, and altogether there are three domains: eukarya, archaea, and bacteria, right? And now we are moving into the basic discussion of taxonomy. So taxonomy, in short, refers to the overall process of classification. And taxonomy actually, it's a practical way of classifying organism. So this is the overall process of classification of organisms by arranging them into groups based on their similarities of large units, right? So based on similarity, organisms are categorized into larger groups. That's what we call taxonomy. And this is done to establish relationships between one group of organism and other group of organism, and also to differentiate one group from other group, right? So we will have a particular group and they will have peculiar characters, and there is the second group, and this group is entirely different from the first group in terms of characters. So in that way we can distinguish the organisms, we can classify, or we can include the organisms into different groups, right? And usually this provides a common reference for identifying organisms which already have been classified. All right. And taxonomy in general consists of three interrelated processes. That means when it comes to the practical side, there are three interrelated processes. One is classification, then nomenclature, and identification. These are actually interrelated. Each are having their own importance and their way of doing things. Okay, so these are the three important interrelated processes in any classification or any taxonomical process. What is classification, nomenclature, and identification? So classification, in brief, let's see what are these three. Classification is the process of dividing organisms on the basis of similarities which they share, right? So based on the similarity, the organisms are classified or divided. This process is called classification. So it's usually used to arrange organisms into a specific group, and these groups are what we call the taxa, right? That we will see later. Then, so this is nothing but the classification, grouping of organisms. And nomenclature is the process by which a name is assigned to a group, right? The process by which a name is assigned to a particular group. All right. And for that, there are certain rules and regulations that we will see later. Then identification is actually the practical side of taxonomy, and in which when you have an isolate, when you have a organism which is new, and these organisms will be identified first. That means we will study the organism, we will observe their characters, we will analyze the features. This process is called identification. And after this identification process, you will get the data, and this data is used to classify organisms. All right. So then classification, nomenclature, and identification both are interrelated and actually very much connected to each other. So we'll start the discussion with the nomenclature first. Right. So nomenclature is, as already stated, is a branch of taxonomy, and in which the names are assigned to a taxonomic group. And for that, we have to follow the published rules. And actually, there is a very commonly accepted rule for classification of living organism, which is the binomial system of nomenclature. And in bacteria also we follow that. This was introduced by Carl Linnaeus, right? So in this binomial system of nomenclature, this may be very much familiar to you. We know that there are two parts for the name. One is the species name, the second part is the genus name, right? And usually it starts with the genus name. So the genus name will be the first part, and the species name is the second part. And a genus name must start with a capital letter, and the second species name should start with a small letter, right? And both the parts of the name should be either italicized, or there should be an underline, separate underline should be given. So this is the idea, or this is the rules for providing a name or assigning a name to a group of organisms. So here you can see the taxonomic hierarchy, or different levels of taxonomic groups based on or which are widely used in classification systems. You can see there is since it is in bacteria, we use the term domain instead of kingdom. You can see there is domain, then phylum, class, order, family, genus, and species. These are the 7 levels of classification, or these are the 7 orders, or a taxonomic hierarchical order for classifying organism. So of these, species is the very basic unit, right? Species is the very basic unit. You can take a lot of examples in this particular slide itself. See, when you take an example like Shigella flexneri or Shigella dysentery, this is a species. So here Shigella dysentery means there is a genus name Shigella, and dysentery is the species name. So this dysentery species belongs to the genus Shigella, and Shigella belongs to a family called Enterobacteriaceae. And this Enterobacteriaceae family is a member of the order Enterobacteriale, right? And this Enterobacteriale belongs to a class Gamma Proteobacteria, and this class is a member in the phylum Proteobacteria, and this Proteobacteria belongs to the domain or the kingdom Bacteria. So this is the way how do we classify organism. So when we go for an identification process, we have to include this organism in all these levels, right? We have to identify these levels of organisms, levels of this unknown organism. Fine. So we can define all those things. Species, actually we'll define that later. So suppose when we define a particular organism, and each organism will have all these things. That means each organism will have a species, will have a genus, there will be family, order, class, and everything will be there for each and every organism, right? So in this one, a particular hierarchical class, hierarchical order in which the organisms are classified, and there will be classes above and below of that. For example, if you take family, right? If you take family, so family is a is a collection of organism in which similar organisms are classified, right? And there will be, when you go up, that means when you go up, that means to the order, the order consists of different families which are related, right? So order is having more number of organisms than the family. And when you come down to genus, genus contains, this is a particular genus, right? That means this genus belongs to this family, and this family may contain different number of genus, right? So when it comes down, the number of organisms are reduced. Okay. So in a prokaryotic system, the definition of species is slightly different. In case of a higher level organism, we know that this is a group of organisms which are having similarity, and also they are interbreeding, right? They are sexually reproducing and interbreeding. But in case of prokaryotes, we know there is no such sexual reproduction, and hence a prokaryotic species is defined as a collection of strains that share stable properties and differ significantly from other groups or other groups of strains, right? So species is a collection of strains with similar properties. That is what a prokaryotic species. It could also be defined in other ways, like these are having G plus E content of similar digit, right? So they have a similar G plus E composition, or it could be having a similarity of more than 70 percentage when you take an overall similarity. So in all those ways, you can define a species. And a taxa or a taxon represents all these subdivisions used to classify organisms, just like we have seen the different levels of classification, hierarchical levels. So domain, kingdom, phylum, family, genus, everything, all these things are the taxa of classification. So species, we defined it as a collection of strain. So what is a strain? Strain is a population of organisms that's distinguishable from at least some other populations within a particular taxonomy category and considered to have descended from a single organism or a pure culture isolate, right? In simple sense, it's a pure culture, right? It's a pure culture of strain or pure culture of organism in which there is only a single type of bacteria. Usually they have been originated from a single cell, right? So a pure culture is often a clone which is having a population of cells derived from a single parent cells. That actually we know, just like a colony, it's a clone, right? And all the cells within that clone will be identical, isn't it? And even it is so, by definition, all these organisms are identical. When you analyze them in depth, when you deep dive into the features of a strain, you can see certain differences. And these differences within a strain are categorized as biovars, right? Or their variants. All right. And within a strain, you can see variants like morphovars, serovars, and these are actually, in general, biovars are variant prokaryotic strains characterized by biochemical or physiological differences. All right, this is the general term used to denote the different organisms within a strain, right? So strains are the very basic unit. That means even within a species is the basic unit of classification, that already we have seen. And a species actually is a collection of strains, and strain is a group of organism. They have been descended from a single parent cell, right? And even within that clone, you can see certain differences between the organism, and that organisms are called biovars, right? And these biovars are of different types, like morphovars and serovars, based on which feature they shows differences. If they shows a feature difference in morphology, it's called a morphovar. If they have difference in antigenic properties, then they are called serovars. And one more point about a type strain, right? When we talk about the classification systems, we need to, when we identify a particular organism and we study that organism, we got certain data, then we need to classify this organism, or we need to compare this organism with an existing organism, right, or an existing group of organism. And for that we need a template, right? We need a strain, we need an organism to which we can compare our unknown organism. And these type of organisms are called type strain, right? So in every group, in every taxa, there will be a particular type strain that is the most representative member of that particular group, right? So this is the most characterized and usually the first isolated strain within a species, right? So this may be considered as a reference organism for that species. And there are different organizations like ATCC and MTCC where these type strains have been collected. ATCC represents American Type Culture Collection, and this is the premier global biological material resource and standards organization whose mission focuses on the acquisition, authentication, production, preservation, development, and distribution of standard reference microorganisms and other things also, right? So it's usually situated in USA, and it provide the pure cultures worldwide. All right. And there is a similar type of thing in India, which is called MTCC, Microbial Type Culture Collection. It's maintained in IMTech, that means Institute of Microbial Technology, Chandigarh, and they also provide this pure culture or type strain across the world. Even not only MTCC and AT, there are many other culture collection centers, but these two are the very common representative examples I'm taking. So let's wind up this introductory part of taxonomy. In the next segment, we will see different classification system. Thank you, and hope you understood.